Welcome to Getting Started with Primary Cells, Standard Procedures in Cell Culture. PromoCell offers a variety of primary cells. At our headquarters in Heidelberg, Germany, we isolate primary cells and put them into culture. We then cryopreserve with cryo-SFM and send them to you, the researcher, to thaw. Thawing is a very important step in cell culture and the first section of this training. When thawing, the overall goal is to obtain as many healthy and proliferating cells as possible. However, the procedure is stressful for the cells. The freezing media contains DMSO as a cryoprotectant, and DMSO can be toxic to the cells, so the final concentration should not exceed 1%. Therefore, thaw the cells as fast as possible at 37 degrees Celsius. Make sure everything is prepared in advance before removing the cells from the liquid nitrogen tank, and dilute the DMSO containing freezing medium instead of centrifuging the cells. For most of our primary cells, we do not recommend centrifugation during the thawing step. It's also good to avoid any excessive pipetting and mixing. When starting your thaw, it's important to calculate the plating surface, and this depends upon the number of viable cells in the vial as well as the optimal plating density for your cell type. These two values can be found in the certificate of analysis for the number of viable cells and in the product manual for the plating density. In this example, we're going to look at human chondrocytes. The optimal plating density is 10,000 to 20,000 cells per centimeter squared. So in this example, in a vial with 500,000 cells divided by 10,000 cells per centimeter squared, the optimal plating surface would be 50 centimeters squared. And so then you can go ahead and prepare two T25 flasks, each with 9 mils of medium. It's important to note here that there has to be a minimum of 9 mils of medium in each flask to properly dilute out the DMSO. Before starting the thaw, prepare the cell culture flask with the growth medium. Then place the flask in a CO2 incubator for about 30 minutes to let it equilibrate. Use this time to double check that your water bath is at 37 degrees Celsius. You are now ready to thaw the cells. Take the cryovial out of the liquid nitrogen tank and transport it either on dry ice or in liquid nitrogen. When you get to the water bath, thaw the vial for 2 minutes. Here you can hold the vial by the screw thread cap and slowly swirl it around in the water bath. At this point, the label may fall off, which is fine, and at 2 minutes you can do a check of the vial. If it's completely thawed, then you can go ahead and plate the cells. If there's a small sliver of ice that's disintegrating, again, you can go ahead and plate the cells. If there's a large chunk of ice, you can put it back in the water bath for about 15 to 30 seconds and check it again. Once your vial is thawed, you can disinfect with 70% ethanol and transfer the contents into the pre-warmed flask. You can place the flask in a CO2 incubator, and then once the cells have attached, or the next day, you can perform your first media change. When thawing, it's also important to keep in mind that you should not fully submerge the vial, but instead immerse the vial just up until the screw thread. And also only thaw one vial at a time. We do not recommend to use a styrofoam floater when thawing the cells, or to thaw the vial in your hands. Additionally, if you're taking the vial out of the liquid nitrogen tank, it's okay to go ahead and do a quarter turn to release the pressure and then tighten the cap back up. So in summary, it's always good to keep the cells on dry ice before thawing, to take into account seeding density when you do your thaw, and to dilute out that DMSO instead of centrifuging the cells. You can then go ahead and do your first media change once the cells have attached or the day after thawing. Once the cells are growing in culture, it's also important to do frequent medium changes. The goal of the medium change is to supply fresh nutrients, remove accumulated waste products, and replace any evaporated liquid. The frequency is determined by the cell type and the cell's growth rate. For faster growing cells, it will follow a Monday, Wednesday, Friday schedule, or 3 times a week, and slower growing cells will follow a Monday, Thursday, Monday schedule, or twice a week. To start your medium change, heat up an aliquot to room temperature or to 37 degrees Celsius. Remove the cultures of just one cell type from the incubator and aspirate out the old medium. Add new medium without producing bubbles or scratching the monolayer. Then you can transfer the cultures back into the incubator. If you're using suspension cells, there are two possibilities to change the medium. First, you can increase the volume, so adding fresh medium to the old medium, which is usually double the volume. Or you can do a true medium change after centrifugation. So the old medium is completely replaced by centrifugation, and then you can resuspend the cells in an adequate volume of fresh medium and transfer the suspension into a new culture vessel. As your cells grow, it's important to examine your cultures frequently. Keep an eye on the cell growth, density, and morphology, as well as any evidence of bacterial or fungal contamination. It's also important to not leave the media bottles in the 37C water bath for longer than necessary. Heating the media for several hours on many consecutive days can compromise their quality. We also recommend to examine the medium and other reagents before you use them for any evidence of contamination. The last procedure that we'll discuss is subculture. As the cells grow, the free space gets limited, and they slow down their proliferation. For further expansion, the cells need to be detached and replated onto a larger surface. We recommend to subculture between 70 and 90% confluency, and to use an appropriate enzymatic detachment with an appropriate inhibitor. Suspension cultures can simply be diluted into fresh medium. We recommend the PromoCell Detach Kit for subculture. This kit comes with a buffer solution, trypsin, and trypsin neutralization solution. We first recommend to pre-warm the detachment reagents for 30 minutes at room temperature. You can then aspirate the growth medium from your flask and wash the monolayer with the buffer solution at room temperature. You can then aspirate the washing solution and add in the trypsin EDTA solution at 2 to 3 mL per T25 and 5 to 6 mL per T75. It's important to note that PromoCell trypsin is at a lower concentration, which is more optimal for primary cells. You can then watch the cells detach from the plate under the microscope at room temperature, and incubation time is dependent on cell type and cell density. So let's take a look under the microscope. Once you add trypsin to the culture, the cells will begin to detach from the plate. As you can see, they'll start to round up, and over time, the culture will consist of just the detached cells. Once you have a suspension of cells, you can proceed with the next step. Stop the reaction with the trypsin neutralization solution. This is used at the same volume of the trypsin. Resuspend the cells by pipetting up and down and transfer them to a centrifuge tube. Spin them down at 220 G for 3 to 5 minutes. Remove the clear supernatant and resuspend the pellet in fresh growth medium. You can then determine your cell number and seed cells at the recommended seeding density into a new cell culture flask. Then you can put the cells back in the incubator at 37 degrees Celsius. Accutase is another detachment reagent, and the procedure is almost identical to classical trypsinization. First, aspirate the growth media and wash the cells with the buffer solution at room temperature. Then add the accutase and incubate at room temperature or 37°C for 5 to 10 minutes. Once the cells detach, transfer them into a centrifuge tube and spin down. Resuspend the pellet in fresh medium and determine the cell number and reseed the cells at the recommended seeding density. You can then calculate the plating surface and plating volume after subculture. The plating surface is dependent upon the cell count, the total volume of cell suspension, and the plating density. So in this example, you would have 150 centimeters squared of new plating surface or 2 T75 flasks. The plating volume is dependent upon plating density, tissue culture vessel surface, and cell count. So in this example, you would have half a mil of cell suspension and plating volume. Therefore, you would plate half a mil of cell suspension plus 15 mils of fresh growth medium in each of the 2 T75 flasks. It's important to only subculture one cell type at a time to avoid any cross-contamination. We also don't recommend to subculture the cells too frequently, so it's best to wait until they've reached at least 70% confluency, which is typically every 5 to 7 days. Trypsinization times are also cell type dependent, so endothelial cells, for example, will detach a lot faster than, say, keratinocytes. It's also important to time your subculture, so extended exposure to trypsin can damage the cells. And if you find that the subculture is taking too long, you can always switch to accutase, which is much gentler on the cells. Thank you for your time today, and feel free to contact us with any questions.